Thank you. Um, Let me see if we can get the slideshow going. I wish to thank a friend of mine, Porter Anderson, who's a vaccinologist. He was the one who developed Haemophilus influenza vaccine, and we had a conversation a little more than a year ago regarding the pandemic and why I thought perhaps children were less susceptible to the ravages of the virus, possibly because of the presence of the tonsils and adenoids that children have. And this led to writing a paper that got published in Frontiers in Immunology last September. And as plain as the nose on your face, the case for a nasal mucosal route of vaccine administration for uh, COVID-19 disease. And that leads me to this presentation. I want to thank Porter for introducing me to Preston and your group. In any case, this discussion will be a little different than the one that we've all had up till now. It's a little more scientific. And it's about mucosal immunity, which represents the interaction between the host and the environment. And this is determined by the pathogen's route of exposure and entry. The upper airway is the most accessible and vulnerable to droplets and aerosols containing bacteria and viruses. And the figures on the right basically show the process by which vaccines, I'm sorry, by which viruses are transmitted. There are particular vulnerabilities to the SARS-CoV-2, and this is determined by viral entry genes in the nasal epithelium that lead to a high efficiency of the COVID-19 transmission. Nasal secretory cells show the highest level of expression of the ACE2 receptor in the respiratory tree, which represents a loci of infection, reservoirs of the infection. And this is confirmed by the uh, observation that higher viral loads are found in nasal swabs compared to throat swabs. And again, the binding of the spike protein to the ACE2 receptor allows entry and replication of the virus. And there are other associated molecules that are required for further processing of the virus, which are basically proteases, enzymes that allow conformational changes in the virus and then processing of the virus within the cell itself. Uh, the nasal epithelium is really the first defense in the upper airway. And this is established by the barrier function of a mucus layer comprised of a gel layer that's produced by the goblet cells. And this basically, we all know, is the typical runny nose. There are ciliated cells which produce waves that basically eliminate antigens, pathogens, and that basically ends up causing the typical sneeze. At the cellular level, we find that there are tight junctions which bind the epithelial cells together that again provide another barrier to the viral entry. There are also pattern recognition receptors that are expressed in this epithelial tissue, toll-like receptors, and pathogen-associated molecular pattern molecules, which basically have a background in terms of a previous exposure to similar pathogens and shared antigens that these pathogens have amongst each other. There is this concept of a gate control that provides, again, the protection, the barrier protection, and this is associated with genes that are expressed by the goblet cells and ciliated cells in the nasal mucosa. There are also the expression of host defense peptides, the whey acidic proteins, and other enzymes which can actually have an effect on the degradation of the pathogens even before they get into the body. The nasal-associated lymphatic tissue is the largest, is part of the mucosal immune system, which is the largest lymphoid tissue in the body. In humans, it's known as the Waldeyer's ring, made up of tonsils and adenoids. It represents the inductive and the effector sites of immune cells, and all immune cells basically are found in this mucosal environment in the nose and in the throat. antigen presenting cells, monocytes, macrophages, T cells and their subsets, and B cells. And the diagram on the right basically shows a very complicated and complex area of this area of mucosal immunity. And it represented an integrated immune response with the identification and processing of antigens, the differentiation of to memory and effector B cells and plasma cells, which are responsible for the production of secretory IgA antibodies for the most part, as well as the pentameric IgM. There's also the presence of a receptor, which allows for the expression of these antibodies to the mucosal surface. And the diagram on the right again shows you the types of antibodies that are produced. The production of in the nasal mucosa and as well as in the tonsillar mucosa, IgA antibodies are produced both locally and systemically, which is important and to be differentiated from the intramuscular approach, which basically just produces a monomeric IgG response. As we see on the right, the IgG basically just has two arms, whereas the IgA can be a dimer, a tetramer, or a pentamer. And this has the advantage of being able to attach to antigens and pathogens that provide for this concept of immune exclusion, and where the secretory IgA is the most abundantly produced antibody. Its multimeric structure basically contributes to the non-specific binding to viral peptides, and this can provide for exclusion at this level. And this is important actually to keep in mind as we go through this talk, that variants are appearing, and the antecedent production of antibodies to the wild type virus that was to which the vaccine was originally developed can lead to this non-specific production of antibodies that may not be specific to the new variants, but actually has been able to identify elements of the variants and then allow for the exclusion of the new variants. The advantage of the IgA antibodies is that they do not activate any of the complement-mediated inflammatory pathways. They also block the IgG and IgM complement-mediated inflammatory effects, and they limit the recruitment of neutrophils and inflammasomes, which are important in terms of the neutrophil extracellular nets that basically recruit neutrophils to the lung and are an active component to the process that causes pneumonia. IgG antibodies activate this complement-mediated inflammation, and then may end up become involved in the process of pneumonia. A paper came out this year that shows that the IgA antibodies dominate the early neutralizing antibody response to the SARS-CoV-2 virus. This is a humoral response that is an antibody response, and they're detected earlier. The IgA antibodies are detected earlier than the SARS-CoV-2 specific IgG antibodies. The neutralization by the IgA antibodies was more potent than IgG in the early phase of the infection. However, in the convalescent serum, IgA was twofold less potent than IgG. Again, looking at the bronchoalveolar lavage, that's basically taking samples from the lungs, IgG concentrations were higher in the BAL than the IgA. However, the IgA antibodies were detected in most of these lavages, whereas the IgG was only found in half the samples. The IgA concentrations in the saliva were higher than IgG and persisted for longer periods of time. Mucosal defenses are essential and critical. IgA antibodies reduce the infectivity of secretions, and they reduce transmission. A preprint was recently released in Med Archives from the University of Florida, which reported that current vaccines reduce transmission infectiousness by only 54% when vaccinated people are infected compared to unvaccinated people who became infected. Another preprint from Israel showed a fourfold reduction in viral load in vaccinated people that would lead to a decrease in the rate of transmission. However, that paper did not address the issue of infectiousness transmission by the vaccinated population. However, it was implied that the reduction by fourfold viral load would actually contribute to the decreased transmission. Vaccines are, as we all know, prevent morbidity and mortality. Historically, they were administered by mucosal surfaces, first reported in China around 1000 AD in a manuscript that was written either by a nun or a monk that showed the dried powder administered by nose actually prevented the further disease process. This may have been picked up by a Chinese emperor in the 16 AD and who gave smallpox particles by insufflation that protected his entire family. This was again repeated in India and in the Ottoman Empire and eventually reached England and New England by the 1700s. Smallpox vaccine, as we know, was administered by scratching the skin. And ideally, vaccines offer lifelong immunity against infection, transmission, and further progression of disease and death. In terms of the vaccine development for SARS-CoV, more than 40 countries and regions are involved in the development of a vaccine. Since February 2021, 256 COVID-19 vaccine candidates have been developed. 96 are in clinical trials, 16 are in phase 3 or 4, and there are 8 intranasal vaccine clinical trials that are ongoing. The figure on the right shows the distribution of the vaccine candidates among the top 7 countries regarding the various stages and the vaccine platform that's being used. The vaccine platforms have increased with the SARS-CoV-2 development. Inactivated vaccines provide a broader spectrum of antigens because they're processed by the immune system based on the various epitopes that are identified through the processing of the inactivated vaccine. Protein-based vaccines provide a more targeted approach. Viral vector vaccines deliver antigen encoding DNA to cells to enhance the inflammatory response. And nucleic acid vaccines serve as a template for the transcriptional translational production of antigen synthesis. There are logistical considerations of an IM versus a mucosal route of administration. Currently approved vaccines require a strict cold chain management, limits its use in rural or in developing countries. Vials, syringes, and needles are required, which can lead to accidental needle sticks and medical waste considerations. Trained healthcare providers are required. 1 or 2 doses present another challenge. 2 doses require that an individual may have to travel great distances in order to get the second vaccine. And the current restrictions are basically age-related. Children under 12 are excluded. Nasal administration, on the other hand, basically require only a single dose. If there is a need for a booster, it can be applied more easily. It's more suitable for children, may be self-administered, and may overcome vaccine hesitancy. The list on the left is quite long. I highlighted Oxford's AstraZeneca Covishield because it's basically taking the same vaccine and they're in clinical trials using the nasal approach. Similarly, for Merck, it switched its failed IM vaccine to a nasal delivery. Dr. Swaminathan, the chief scientist at WHO, believes that the nasal vaccines may not be available until early next year for the youngest of the pediatric population, i.e. Children from 2 to 12 years who are currently being left out of the vaccine delivery trials. Adjuvants are required to promote antigen uptake, and increase antigen presentation, recruitment of immune cells, stimulation of the innate immune response, and polarizing T cells along the type 1 versus type 2 types, also to promote growth and differentiation of B cells. Adjuvants that are available are aluminum salts, which are used for intramuscular injections, natural products from squalene, polysaccharides, and cytokines, synthetic compounds of the lipophilic immune stimulating complexes. The leading one is saponin, which is the derivative from a bark of a South American tree, and the imidazola quinoline derivatives, which are used to stimulate the immune response. Bacterial subunits, the cholera toxin, the E. coli heat label toxins have more or less been abandoned. However, there's interest in a new bacterial subunit that's been developed by John Clements from Tulane. Vaccine delivery also depends on the delivery molecules, the lipid plasma membranes that can overcome the issues involved with the endocytosis of a vaccine. Lipid plasma membranes are used, and there's concern about whether or not they will bind to serum proteins, whether or not they'll be picked up by immune cells prior to their arrival at their desired target, and degradation by nucleases and RNases. Nanoparticle based delivery is certainly at the forefront to promote membrane fusion and promote cargo release. And there's mosaic nanoparticles which can deliver 8 distinct different receptor binding domain peptides. This is basically state of the science. Over 4,000 articles have been published related to COVID-19 vaccine-related research. And the figures on the right basically show by research area and by platform. Protein subunits have received the most attention, and closely behind are the mRNA vaccines. There's particular interest in the cross protection by other vaccines. This is controversial and inconclusive, with a focus on the BCG vaccine. However, current countries with recent MMR vaccines have had fewer COVID-19 deaths, and this is related to the sequence homology between the SARS-CoV-2 glycoprotein, correction there, not one, with the measles virus fusion protein and the rubella virus envelope glycoprotein. There is concern about mutations associated with the S protein. These mutations lead to higher transmissibility, greater virulence, and can lead to the loss of neutralizing activity. There is cross reactivity with the existing vaccines, and the benefit of the mRNA type platform is that an adjustment of the vaccine sequences can be made readily. Special considerations and concerns are antibody-dependent enhancement. This is anamnestic response based on an encounter with similar or same antigens. 90% of the human race has tested positive for 3 of the 4 common cold coronaviruses. Suboptimal vaccines may produce non-neutralizing antibodies of the IgG isotype that may enhance infection to the wild-type virus as well as to variants. And there's vaccine hesitancy concerns. Also the concern for durability of the immunity and immunosenescence in the aging population. However, it was pointed out in one paper that the gnawed and aged mice was preserved to a greater extent than the gastrointestinal associated lymphatic tissue. Herd immunity, I'll end on this. The IgG isotype contributes little to no mucosal immunity. The nasopharyngeal tissues are reservoirs for the virus where little or no IgA is expressed. And my question to all of you is, can herd immunity be achieved if transmissibility remains at 54% with the current parenteral vaccines?